Hi, I'm Pranjul Shah.
And I'm Paul Wilmes.
We're a team of researchers, engineers and entrepreneurs from the Luxembourg Center for Systems Biomedicine at the University of Luxembourg.
We, as well as our team, are delighted to have been awarded 2 FNR Awards, one for Outstanding Scientific Publication and another for Outstanding Research-Driven Innovation. These awards are awarded for our artificial gut model called HuMix.
HuMix is an artificial model of the human gut. It consists of 3 different chambers where we cultivate human and bacterial cells, allowing them to communicate across semi-permeable membranes. It is a means to study how human and bacterial cells interact in the human gut in conditions representative of the gut.
Well, our lab works on the human microbiome. The human microbiome is the collective of all microorganisms in and on the human body. The microbiome has currently attracted enormous research interest worldwide because it has been linked to a number of different diseases, including cancer, metabolic diseases and neurodegenerative diseases. With HuMix, we're able to now address a fundamental question: is the microbiome a cause or a consequence in relation to these diseases? And this will have important implications for the future in how we manage our health and disease.
In our publication, we demonstrate how HuMix overcomes majority of limitations of existing systems. For example, HuMix allows cultivation of bacteria without oxygen, which is necessary for most of the bacteria living in the human gut. Additionally, with HuMix, we can study the interaction between the human and bacterial cells, especially coming from human origin, which makes it much more relevant than animal models.
Well, we receive tremendous interest from food and nutrition as well as from pharma companies, and they see the human microbiome as an area for great market growth, and they are of course interested in developing new nutritional products as well as drugs, and it's here where HuMix comes into its own by providing a platform for developing new nutritional products as well as screening for drugs before these would be tested in human clinical trials.
HuMix is a very complicated system, so we had a lot of challenges right from the start. For example, we could not use the standard cell culture medium with antibiotics because that would kill the bacteria. So we had to develop a whole new co-culture protocol along with the device. But now that we have this protocol set, we could use that with multiple devices in parallel for a large number of applications.
So HuMix was really a team effort. It has involved around 17 people working together over the course of the last 6 to 7 years. This involves people here at LCSB, as well as our collaborators at the Luxembourg Institute of Health, as well as Frederick Sennhauser's group at the University of Arizona. So research, of course, takes a long time to generate impact, and it's in this context that it's important to stress the sustained funding through multiple grants from FNR that have really allowed us to take the model and develop it into its present form and hopefully beyond. Well, we are currently in our lab developing the technology further. We are, for example, incorporating additional human cell types in the model with the aim of developing proper personalized models, which is in fact a requirement of our various different industrial partners first and foremost. And it is with them that we're engaged in additional projects, most notably recently funded by an FNR Proof-of-Concept grant, and this will allow us to expand our already extensive patent portfolio to really further mature the technology into a commercial entity.
We're currently discussing with potential investors and soon may have a HuMix-based startup. Ultimately, it is our vision to become a dominant player in the global microbiome therapeutics market with a product which is born and bred in Luxembourg.